was like, that was more of a premonition because that's exactly what would happen.
I assume you want me to work so you can bail earlier, right? That's the concept I'm picking up here. All right, so let's go through. There's just a handful of slides you have to know for this lab. So the repeats of the ones you learned from last lab, just different vocabulary. So you're now supposed to go look at this slide. I know this is a testis, right? Supposed to be able to tell me if these cells are primary or secondary. So from biology 112, what made the cell primary? No clue. It was diploid, and you're going to have 2 sets of chromosomes. A secondary cell, therefore, is what? Haploid or a half set. It's gone through meiosis. This is a meiosis lab. So if you remember that your gametes are meiotic, right? So you're going to start on the outermost ring of the seminiferous tubule. These funny-looking black cells, they're going to make sperm. So they're diploid and they're spermatogonia. They're the initial ones. They're going to divide and make a copy, which is the first row you see. Don't look at him. There's the first row you see that looks round. Those are diploid sperm cells. So how would you say diploid sperm cell in doctor speak? Primary spermatocyte. So the first row down from the edge are your primary spermatocytes. They're still diploid, but they're going to be sperms. Yes.
So the dark ones then are the spermatogonia?
Yes, so gonias are the very outside. They're kind of flat, kind of odd looking. But then the first full round ones are your primary spermatocytes. Then they undergo meiosis. So these rows here, 1, 2, 3, these 4 layers here. If they're in meiosis, they must be what? Secondary spermatocytes, because they're meiotic, they're haploids. So on this picture down here, this flat cell would be my spermatogonia. It's going to make sperm. That thing is primary spermatocyte because it's diploid. These are all my secondary spermatocytes because they're haploid. And the guy with the tail at the end is a what? Spermatid, meaning they're immature. Or spermatid, you can say either way. So if you can look at that slide and walk yourself through gonium, primary, secondary, spermatid. That's the logic you're supposed to know of that slide.
So what's going to identify as secondary?
They're not the outside row. It's the easiest way to do it.
It's like a thicker layer.
They're slightly different looking. They're more than one layer. Or you just count from the inside up. If you go from the lumen, they're the layers before you get the end. But they always look a little fainter, but not always. Yeah. So the primaries are always the normal looking cell along the outside, but not the very outside. These, those are primaries. Secondaries are everything going in from them.
And it's spermatogonia. Is that always going to be like in that?
On the very outside.
Purple layer, it's like
The very outside. It's always that one touching the testis barrier. So you get that logic? So you just count the rings, basically. You can get it.
And the sperm is the stuff in the middle.
Spermatids are the stuff in the middle. The guy with the tails. So if I do a different view, that's going to give you a different view. Because I can give you a different view. Oh, I don't like that. Let's change views here. Let's zoom out a little bit. So if this were a test next week, and I was evil enough to point to this cell here, you would say that's a what?
Secondary.
Secondary spermatocyte, because it's not the outside, and it's not the guys with tails, it's somewhere in between. So that's the secondaries are everything between the outermost layer, basically, and then the spermatids.
So what's that?
So if I were at where this arrow is, right here, how convenient, the arrow of this one, on the very, very outside, it's kind of flat. The spermatogonia, very good.
and primary.
Primary would be this cell here, this row right here. The ones that are touching the gonias.
What about where the pointer for the computer is?
Right from the computer is. Oh, right here? Spermatid.
Okay, so
Nucleus spermatid.
that kind of stuff that looks sort of like muscles. But
Yes. So the nuclei of the sperm are actually over here. They face out. Their little tails go in.
Oh, okay. And the functional sperm is that like?
That'd be upstream. That would be epididymis on out.
Okay.
So downstream, I should say.
Spermatogonia is diploid or haploid?
It's diploid. Some other stuff. But they don't use primary, secondary with it. They just say gonia. So basically, count the rings in a seminiferous tubule. We'll do one more, just one more. So there's a close-up. So if it were me, I'd say, well, this is going to be a gonia. It's on the edge. This would be like a primary. They look kind of big and round. Secondaries, everything I can't quite see. Spermatids are down here. Right? So diploid, diploid, haploid, haploid. Right?
Are there any functional sperm in the seminal?
No, because they'd be after they mature. Okay, on the sperm layer. So basically you're naming the layers. It's not too difficult. The ovary, though, makes life incredibly hard. So what you want to do... Okay, here's an ovary slide. How would I know that's an ovary? It's round, yeah, that's good. And it's got big follicles, the nests, right, where the eggs are. So you have to identify the follicle and the cell in next week's lab, like we kind of did in lecture. So before we do this slide, let's review this poster, which you're going to take a picture of with your phone. Because that is how you do ovaries, is that poster there. So this poster shows you how the ovaries, the follicles, and the eggs are changing size. And the names don't correlate. Just because it's evil. Alright, so let's go back to this slide. You're going to find the smallest round thing on the slide. And start there. Let's go, that'll be right up here, these little teeny round things. So those are baby eggs and a baby follicle. What word would mean the earliest ones? Primordial follicle. What oocyte is in the primordial follicle? Primary, because it is diploid. So I have a primary oocyte in a primordial follicle. So names don't match. Right? Then you're going to go and find some that are slightly bigger and round, like these, which are just a circle, big circle, or these. What would we call a bigger follicle?
Primary.
Primary. And what kind of oocyte's in a bigger follicle?
Primary.
A primary oocyte, because it's still diploid. Right? Then you look for one that's not only big, but then has fluid on each side, like this one here. There's the follicle with fluid around it. So that's a secondary follicle with what kind of oocyte? Primary, because it's still diploid. So far these have all been the same oocyte, but the follicle is changing. Then you're going to look at this ginormous round thing. There's actually an egg stuck on the little end right there. So what kind of follicle is the ginormous round one? Mature or Graafian or vesicular. And what kind of oocyte is in that? Secondary, because I went through meiosis, I'm doing meiosis, so I'm haploid. So the only follicle that has a secondary oocyte is the mature follicle. All the others have primaries. But there is no secondary follicle. The secondary follicle does not have a secondary oocyte. So the follicle naming is separate from the oocyte naming, yeah.
Can you point to some other secondary follicles?
Well, this is one. Smaller one. This is a good one here. Got the egg right there. So all the big ones you see are secondaries, which are mature, not secondary
So
follicle.
like almost kind of overlapping.
So a secondary follicle would be usually smaller than that, not as big. So this is mature. I misspoke. This is mature. Secondary. I'll show you one, actually a better one. I'll zoom in on a secondary versus mature. And you'll see the difference. The difference is size and where. Where's my ovary? Yeah. Here's a close-up. So let's go back to it again. So, tiny, round, indescript place. Which one's those? Primordial follicle with a primary oocyte. Once you get a circle like this, where I can sort of see a ring, you're now a primary follicle with a primary oocyte. A secondary follicle is larger than that and begins to have fluid. So here's a beginning of a secondary. This is a later secondary. Here's a really early secondary. So you start getting fluid on one or both sides. So that's a secondary follicle with a primary oocyte. The big beast with the huge antrum with the egg at one end, that's the mature or Graafian or vesicular. The antrum's the big space. Okay.
Can you point to the antrum again?
The big space where the fluid would be. Anterior antrum. Right, so you're just naming the follicles and naming the cells inside of them by size, basically. Again, this poster is a very good linear picture of that. But then we have a problem. Because after you ovulate, what do you form? That's how your exam later today. Right?
What kind of fluid is in the
First. And
It's called antral fluid or follicular fluid. Basically, it's a saline suspension. So let's go through this again. What is this over here? That's a mature follicle. The egg's on the end. Mature follicle grafting with the antrum. This ugly-looking mass of material, that's where that used to be. But now it's ovulated. So what is that thing called? That's the corpus luteum. That's what this became after the egg left. It becomes this. They're always huge. Ginormous.
Are they always weird shaped?
They're always, yeah, usually. They're kind of fluffy, cloud-like.
Do they shrink in mass?
They'll shrink, and then they get really, really small and become an albican. But that's a corpus luteum. You're going to see a big, puffy-looking area. That's going to be generally bigger than you expect it to be. Because they make a lot of hormones. So let me see if I have it as one. Does anyone find a ginormous corpus luteum in this pregnant ovary? That's this.
Wait,
This,
what is that?
that's a pregnant. If you're pregnant, that's how big they can get. But that's the corpus luteum. Your eggs are all these little tiny things over here. So it can take up the whole ovary. But that's one follicle that went mad. Ovulation, basically. Forms that puffy cloud-like layer.
Is that typical or not typical?
In pregnancy, that's typical. Not in every day. Let me show you another one then. Hey, this is an old corpus luteum. There's actually two of them. There's one here and an older one here. And notice how they're getting much smaller now. So we would call them, they kind of break down and look white. That's the albicans. So this will be shrinking. This will be shrinking more, shrink. But it's not as puffy. It's not as big. It just looks generally smaller and less pronounced. It's a judgment call. If I show you another one of albicans. Here's an albicans. It looks like a small cloud. Okay. So you get your bearings. Here's a primary oocyte. So they're getting much smaller. It just still looks a little odd. It's totally by judgment size. Corpus luteum are huge. Albicans are not.
Where do you tend to rest when they're completely done?
They tend to become this. They tend to move inwards.
And then are the eggs more on the periphery?
Yeah, the eggs begin in the periphery, and the albicans and the luteins are on the side. So here's another one. Here's actually one up here too. There's an old one right here. But those are various albicans.
Can you put an albicans and the other one next to each other? Or one after the other?
I can try. Well, there's one actually both next to each other. Let me see. There might be one. I think they actually have females. Yeah, I don't know how that actually shows side by side a luteum and an albicans. These are albicans. So all of these are dead clouds. They're just a lot smaller. A luteum would be much bigger. I don't have a slide that I think has one by each one.
Well, it's not dead tissue, is it?
Well, yeah, it does kind of necrosis down, and then it scars over. It becomes basically fibrous tissue.
Oh, wow. So it is scar tissue.
Yeah, so if you count that, she ovulated 5 times on this slide. So you can go through and count that. That would stay behind your ovary like that. Right.
So can you count? Yeah, what's the fifth one?
Like 1, 2, 3, 4, 5.
Oh, okay. And those white things in the center of 2 of them?
That's just a blood vessel, yeah, a cavity. So those are albicans. Okay, so then the fun begins on the uterus, because you're all supposed to know the phases of the uterus. So this is the uterus, right? Actually, no, it's the uterus, besides I just told you. Got the glands, got the edge. Okay, so now you're supposed to name what stage the uterus is in. So what you're going to do is look at these glands and mentally notice how they look. So here the glands seem kind of tall. They sort of seem regular. They're all round or tall. Compare that with your mind's eye to this picture here. What do you guys want the glands here? A whole lot of them, and they're squished, right? So the prior slide was as your uterus is growing before ovulation. Go back to this one. So before ovulation, I have fewer glands, and they're elongating because I'm growing my uterus. That's called the proliferative phase. I'm growing, I'm proliferating my uterus.
So you said it's premenstrual?
Pre-ovulatory. Pre-ovulation. Proliferative. And this one is after ovulation. So I'm secreting. That's where they get the name secretory phase. But I have a lot more worms, we call it, and they're packed together and they're coiled. Hence why they're kind of bent. So if they're long or round or even, you're growing a uterus, proliferating. If they're all bundled up, then you're after ovulation, so you're secreting. This is the vascular phase.
Wait, I thought that was the secretory phase.
Yes, that's when you're vascularizing your uterus.
Oh.
Right. So if we go back here. Let me show you those again in a different view. So this is a slide next week. You look at the worms, glands, and say, they all seem kind of even. There's not too many of them. They're round or long. Which phase would this be? Proliferative. This is before you ovulate. Just a normal everyday uterus.
Is the secretory also pre-ovulative?
Nope, it's after ovulation. This one would be secretory, because notice the worms are much more crooked. They look laser-like, you know what we call them. There's more of them. They're crammed together. They're changing shape. This is when you're vascularizing after ovulation, so it calls the secretory phase.
Can it also be called vascular phase, or is it?
No, secretory phase is the only word for the uterus. That's what's happening in the menstrual cycle.
Would you be able to go over a couple more slides?
Sure. With the uterus?
Yeah, the proliferative.
Sure. Let's see if I can find another one here. What is this one? Proliferative. So the trick is the glands are either round or long. They're not really bent. There's not that many of them. So this is when the uterus is growing. So proliferating. And you have to compare that with your mind's eye. Which there's more of them. They have these jagged edges. They look bent, crooked, torn up, right? So a lot more busy. That's the secretory phase. And let's say you're not pregnant. What's the next thing that's going to happen to your uterus? Menstrual phase. So the trick of the menstrual phase is that if you look in the lumen of your uterus, there's pieces coming off. Right here. Right? That's a blood clot. Right here. Right here. Because it's degrading, and so the worms are basically falling off, and then you'll start again with proliferative phase again. You see your chronic tissue.
Is there always going to be stuff in the lumen when we look at these slides?
Almost always, or you'll see where it's all torn up on this outside edge. And that's in the lumen, you'll see it all torn up. Let me show you another couple of views of menstruation. I don't have another menstruation. Okay. I don't think I have another menstrual one. Gosh, where's menstruation when you want one? Let me There's another little small menstrual, because I can see that there's a lot more destruction appearing in the lumen. So I'm seeing holes in the lumen. I'm seeing lots of blood forming out. I'm going to see a lot more destruction. So your three phases of the uterus are those three. Let's try a little range of tests, shall we? Let's do this one here. All right, so I'm going to point to a cell. I'm going to point to this one. And you're going to say?
Secondary. Something.
Spermatocyte. Right, very good. Because the spermatids are there. So haploid sperm forming cell. Secondary spermatocyte. Let's then do. Hey, if I pointed to this, what would that be?
Secondary.
Not secondary yet. Primary follicle, it's a ring. Right.
It's not fluid around it though.
There's no fluid around it. They're just seeing the cell in the center.
So where's the egg in that?
That is the egg.
So is it just like 2 layers?
Yes. So let me show you another view here. So if you zoom in on a primordial follicle, all you see is the egg. You'll see a random cell like this. That's a primordial follicle. It's a very thin follicle with an egg in the center. So primary oocyte of primordial follicle. Once they get a ring around them, like this, that's a primary follicle. Now I have a thick ring. This is the egg. Here's the ring around my egg, but there's one ring around the egg, basically. Primary follicle. If I get fluid on I start getting fluid around the follicle, like over here and over here. As my follicle gets a space around it, now I'm a secondary follicle. I'm getting fluid around my follicle. And then I do this one here, for example. That's the mature or Graphian, is when the egg's on a stalk, and you have the antrum around it.
Like a heart.
Yes.
Antrum, like a little lumen.
Yeah, it's like the lumen of the follicle.
Just the white.
Well, it's the white and the red. The red's fluid inside the antrum.
And it's pinkish one?
Yes. So this whole thing's the antrum. The whole thing. That's the egg.
That's the oocyte?
That's the secondary oocyte.
The secondary oocyte.
Yeah, because you finished you started meiosis, so you're haploid. The trick with that is looking for the follicle. Then we have, let's try this one. What stage is this slide?
Secretary.
Secretory. Lots of glands are all different shapes and bent. They're all crammed together. It's the secondary secretory phase of a uterus. Makes sense? Those are the 3 slides you're going to get. Testis, ovary, and uterus. Those are the 3 things you have to be able to look at and see what you're seeing. Yeah, that poster is really good. In addition, ovary. I think I did this way. So you have those basically 3 kinds of slides. In addition, you're supposed to know meiosis again. If you forgot meiosis, shame on you. If you go to your objectives, there's some vocabulary on page 24 you're supposed to remember about a meiosis. Crossing over, dyad, diploid, zygote, blah, 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 blah. We could ask you to go through the meiosis stages again, explain which ones are haploid, which ones are diploid, is it a bivalent, is it a tetrad, all that stuff in one swell. You can even do the meiosis square dance if you want. You ever heard of meiosis square dance?
No.
Oh, it's so great.
No.
I'll sit here and just dance along with it. Okay. Basically, those are your jobs, is to know those slides. In addition, we have one model for you, which everyone last week thought was a giant breast. It was not. This is an ovary. Yes, I can see the nipples. When I'm a male, I see nipples everywhere. But this is the ovary, and so it has the corpus luteum and everything on it you can identify as well. So this model and those slides is basically what's on your quiz next week. So a lot of paper pencil. Every meiosis. Yes! This is my favorite. PMAT times 2. PMAT times 2. That's all. So take a picture of that. Use the rest of your time as you see fit. Or not.
How do I do this?
You can take it down. Nick, you buy those disposable cameras. Most weddings have them.